both large-sized and widely distributed throughout the lake, and so the two where the cutthroat trout are. Now, when we look down to almost all those, the trout were cutthroat trout and very few of the lake trout. Not terribly surprising. Okay, the other question from the earlier work was the interest in otters and transporting nutrients between water and land. And so for this, we turn to doing stable isotope analyses. And so I'm going to give you a bit of background on that if you're not familiar with that. So we can look at the ratios of the heavy isotope, the nitrogen-15 isotope, to the lighter isotope, nitrogen-14 isotope, in different types of samples. And this gives us a way to kind of trace the origin of where that nitrogen came from in a sample. And in these cases, the samples were tissues of plants that we collected on otter latrine sites. So to give a bit of a cartoon of what I mean by this, if we focus just here on, this is going to be a y-axis of increasing nitrogen-15, that heavy isotope. Nitrogen-15 tends to be higher in the tissues of organisms as you go up a food chain. So there's higher levels of N15 in an owl than down here with this mouse, something like that. So it's just a general idea that it increases as you go up the food chain. Now when these guys poop or pee out some nitrogenous waste, and a plant takes that up with its roots and takes it into its tissues, it'll incorporate that signature. So that means if this mouse was to pee on this plant and the plant takes up that nitrogen from that urine from the mouse, that will be a lower level of N15 than if a mink pees on that plant. Okay, so it gives us an idea of the source of where that came from. So this is kind of important. I go through that a little bit for some of the stuff I'm going to show you in a minute. Does that make sense? I don't know. Okay, so again, predator excreta are usually enriched. They have higher levels of N15 than what's in the background kind of soil nitrogen. So when we pick vegetation samples from different plant species on otter latrines and then compare them with non-latrines or reference sites, plant samples off of an otter latrine, we see that N15 levels are higher really kind of across the board in the plants from the otter latrine than from the non-latrine site. So this does suggest that yes, these plants are taking up nitrogen made available. They're fertilized by nitrogen made available by otters through that scent marking activity. We also see that those levels of N15 seem to be higher on the sites, in plants on sites that have higher levels of otter activity. So more otters, more active sites have higher levels of N15 in their tissues. So that's kind of what we knew getting up into our more recent stuff. So we've established that it appears that the otters are pretty dependent on this cutthroat trout in the spawning season. Otters do appear to contribute some nitrogen to the terrestrial plants via their marking behavior. But so now the larger question becomes, well, what about what's going on with the otter population itself, given these big declines in the native cutthroat trout population? So the questions, this kind of frames some of the questions that we asked here. So have otters declined since the decline in cutthroat trout? To what extent would changes in the abundance and distribution of otters in this area affect those water to land nutrient linkages? And then we're going to step back again and kind of look at some of the compounding hypoxic effects that otters face, low oxygen availability effects that they face, and see if we can predict anything about their physiological capacity to even switch to preying on those deep water, open water living lake trout, a novel prey species in this environment. I know I move my hands around a lot, especially. I can't stop myself. Okay, so we're going to go to our first section. So we'll go through these three parts, and our first section is going to be the kind of the predator abundance and behavior portion. So this is our river otter population study. So our question is, of course, have otters decline with cutthroat trout? So one of the things right away that hampers us a little bit in this is that we came in and began studying otters, as I mentioned, in 2002, which is at this point when we look at the declines in cutthroat trout. So we've already come in after a big crash in cutthroat trout. So we don't really know what was going on back here because there really are very little historical data on what was going on with the otter population before that time. Also, it's important to note that the impact of some of these stressors, some of these non-native stressors, like the lake trout and the whirling disease, on the cutthroat trout vary kind of spatially throughout Yellowstone Lake. So For instance, Pelican Creek is the second largest tributary to Yellowstone Lake, and it had an especially high prevalence of that whirling disease in it. And during the period of our work on the River Otter project, cutthroat trout spawning numbers had essentially vanished. So they'd gone from highs of 30,000 fish going into the Pelican Creek to nothing. They weren't in there anymore by the end of our studies. So because of that, I'm going to point out in this next section a couple areas, three streams that were especially impacted by declines in cutthroat trout via these non-native species during our work. So I'm going to return to kind of these. There's Pelican Creek, Cub Creek, Clear Creek. So when I show you those, that's where I mean. Okay, so our objectives for this first part, one is we wanted to use DNA analyses. So we collected scats and hair from otters, and we identified individual animals from those samples and used them in a capture-recapture analysis to basically look at abundance distribution of otters in this system. Also, we wanted, because there really are no historical data on otter population size in this area, we just wanted to provide a baseline estimate for monitoring otters in the future. And then finally, because we've collected diet data in 2002, 2003, we wanted to compare the diets over a long period of time. So briefly, I'll just mention that we did our latrine site surveys from late May through August each year. We collected scats throughout the study for DNA analyses. We began implementing hair snares in 2006, and then we used these samples to DNA analyses on these samples to identify individual otters. Same idea as kind of a CSI crime scene deal. If we can identify a hair sample from a criminal, we can identify the individual. That's what we're doing here. We sampled 7 microsatellite loci, and we used the SRY marker to differentiate between males and females, male-female otters in those scat and hair samples. Then we used the individual identifications from those samples in a capture-recapture or mark-recapture framework. And this thing looks a little nasty, but I won't go through the nitty-gritty. But we estimated abundance and survival for otters with open population models, as well as a closed population models that assumes population closure. In the open population model, for those who are interested, we introduced covariates including sex, time, effort, or the number of observer days we were in the field at these otter latrine sites, and as well as cutthroat trout spawning numbers and gillnet numbers from the lake gill netting, the indices I showed you at the beginning of the talk. We calculated otter densities as how many otters as number of otters per length of shoreline that we surveyed. So density is calculated a little bit differently for an otter than you may think of because they exist along a shoreline corridor. We also, given that we'd had this big crash in the cutthroat trout population, we looked for any evidence of a population bottleneck or a big severe decline in the otter population as well by using this bottleneck test, which tests for discrepancy between allele frequencies and heterozygosities. So we looked for evidence of a genetic bottleneck in the otter population that may have coincided with the crash in the cutthroat trout. We did diet analyses, which is a pretty standard thing, just picking through the scats and identifying prey in the scats. We sent a subsample of scats and bones to a professional identification laboratory to assist in differentiating between cutthroat trout and lake trout in the samples, which is by far the most difficult part of these diet analyses. So, some results for our first part, for our population part of the study. Well, we identified 58 different otters over the entire study period, and this map shows you how many different otters were found at some of those different latrine sites that we visited. We found a significant departure from Hardy-Weinberg equilibrium and some evidence of inbreeding in the population, which is not too surprising, and it's been found in other otter studies with especially potentially isolated or small populations. Annual survival for otters was 67%, which is relatively low. However, there are really only 1 or 2 studies that have reported survival for otters, but compared to those, it's relatively low. Recapture rate was 43%, and some of the top-ranked populations, some of the top-ranked models that we had showed some evidence of survival being dependent on spawning cutthroat trout. But these models really produce pretty wide and biologically kind of implausible confidence intervals, and so we ended up using the more parsimonious, simple model of time constant survival in our estimates, in our survival estimates, and abundance estimates. So, some evidence of cutthroat trout dependence, survival independent cutthroat trout. Okay, so when we look at actual abundance and population size estimates, this shows population size over 2002 to 2008. Essentially what we found here was no statistically significant change in otter population size over the period of our study. So although there's some fluctuations within the years, there's essentially no change in population size from 2002 to 2008. However, otter densities are fairly low relative to other systems, so Yellowstone falls out kind of at the low end for otter densities, number of otters per shoreline. And also those bottleneck tests indicated some evidence of a genetic bottleneck, and the power of these tests to detect a bottleneck is often fairly low after only about 10 generations. So based on otter generation time, this would mean that if a bottleneck happened, it would have been somewhere in maybe in the last 30 years or so, not 100 or 200 years ago, but somewhere more recently. So this may suggest that the otter population had already declined by the time we came in here at 2002, which is pretty late in the game as far as when the cutthroat trout have already crashed. So what I've shown you so far is during the periods of our work, we're not seeing a numerical response in these otters to changing cutthroat trout numbers. But we do find some kind of interesting changes in behavior here. So again, I'm going to kind of highlight Pelican Creek, Cub Creek, and Clear Creek, and I'm going to show you a couple graphs of those, as well as the shoreline of the Yellowstone Lake. So this first series is showing you latrine site activity. So again, number of scats per site, how active are the otters at these sites, on Cub, Clear, Pelican, and then the lake shoreline. Over here we have those indices of cutthroat trout from the Park Service for the lake shore and Clear Creek. So what we see really is big declines in otter activity on all three of those tributary streams, even during the period of our work, and a decline on the lake shoreline as well. Some of these, it's not that surprising. I mean, Pelican Creek, there were no more trout in there. They weren't spawning in there, and the otters responded in kind to that. Latrine site densities. This is now the number of latrines that are active per kilometer, so how many latrines are they using per kilometer are on the y-axis here over those same years. Here we see Cub Creek, Clear Creek, and Pelican Creek declining. Not so much of a change on the lake shoreline in terms of latrine densities. We also see some evidence when we look at the distance that otters were found between latrines, or how far they're traveling between latrine sites on our visits to latrines. We find that it seems that they've increased the distance that they've moved in the later years of the study, potentially because there are fewer prey available, they're moving more in search of prey. It's a pretty common finding in studies of otters as well as many predators. As prey availability declines, predators increase home range sizes, increase distance moved in search of prey. At the same time, we find that even throughout the study, cutthroat trout are a seasonally important prey for these otters, even as they're declining. This is distance moved by otters between latrines, and they truncate their movement, they decrease their movement during the peak of that cutthroat trout spawning period and then increase it afterwards. So it seems that otters are still concentrating, reducing their movement and concentrating activity on those few tributary streams where there are still cutthroat trout spawning, or those few cutthroat trout that are spawning. Here are some diet data over throughout the years. We have frequency of occurrence of different prey types, the percentage in otter scats from 2002 up to 2008. Trout declined from a high of 73% in 2003 to 53% in the last years of the study, although they remain the top prey. Suckers show a trend towards increase in their diet, and then especially we see an increase in amphibians, and this was primarily frogs in their diets as we go through the years. When we look at those scats that we are able to differentiate between the lake trout, we have fewer lake trout. Okay, so we'll wrap up this first part and say that we found no significant change in otter population size during our study. However, overall densities were relatively low compared with other regions in North America. There was some evidence of a recent genetic bottleneck that may indicate that the population had declined, by the onset of our study in 2002. And annual survival was relatively low and correlated with cutthroat trout abundance in some models. And although otters did not seem to respond numerically to changes in cutthroat trout abundance during our study, they did show some behavioral responses in terms of their spatial distribution and their diet. And introduced lake trout were infrequent in their diet, remained infrequent throughout. And I thought I would also mention that hopefully what you can see from what I showed you earlier with the diet data is that those suckers, those long-nosed suckers, are by far an important prey for them as well. And those lake trout will eat those suckers too. And it's kind of hard to envision a scenario where if the sucker population went down for any reason, that wouldn't be pretty bad for the otters, because there's not a lot else for them to eat after that. So, okay, so I'm going to switch gears pretty abruptly here and turn to more of a physiological study. And so here we're interested in, and we're going to tie it into the lake trout, cutthroat trout issue as well, but we're interested in what happens to an otter in Yellowstone Lake, because they are a diving species that occurs at high elevation in Yellowstone Lake. So this is the high altitude diving portion of the study, which is what Hank worked on as well, and this is what he mentioned at the beginning. So some background on this: if you're a high altitude diver, you face two forms of hypoxia, or low oxygen availability. One is at high elevation, there's lower partial pressure of oxygen, as everybody's aware, and also you face a dive apnea or lack of oxygen when you're holding your breath. So you're facing those two forms. There's a really pretty rich body of literature, physiological literature on how organisms cope with extreme environments, extreme hypoxic environments. A lot of work on, of course, humans at high elevations, such as Tibet and in Andes. A lot of work on really extreme deep diving species, such as like a sperm whale can hold its breath for an hour and a half. So like really extreme deep divers. But there's not really a lot of looking at what happens when you put these two things together. And there's a little bit of work on burrowers at high elevation, such as these naked mole rats. They face low oxygen availability in their burrows, but then they're also at high elevation. But really nothing on what happens if you're a high altitude diver, if you put those two together. So we'll look at a couple mechanisms that you can use to cope with hypoxia. And so I'm going to spend a bit of time on this slide to hopefully not confuse everybody, including myself. But one thing that you can do, one mechanism for coping with hypoxia is you can change your hemoglobin's affinity for oxygen. How readily does it take up oxygen or release oxygen? That's affinity. And this is often looked at on this thing called an oxygen dissociation curve, which shows the percentage of hemoglobin in the blood that are saturated with oxygen over increasing partial pressure of oxygen. And so as blood flows through the body, partial pressure of oxygen is very high at the lungs, and so that would be right around here, and that means most of your hemoglobin is saturated with oxygen. As your blood goes out to your tissues and deposits and reduces oxygen out to the muscles and the tissues, partial pressure of oxygen is lower, hemoglobin's unloading oxygen, so you're down here in this area. You've got lower saturation of hemoglobin. Okay, so the affinity for hemoglobin is often reported with this thing called a P50 value. And all that means is what is the partial pressure of oxygen at which your hemoglobin is 50% saturated. 50% of the hemoglobin is saturated with oxygen. And I mentioned that because I'm going to show you a few P50 values, so I just want to kind of show you what I mean by that. Okay, so there are several mechanisms that can cause your hemoglobin to increase its affinity, more readily suck up oxygen. And what that does is it takes this curve and it effectively shifts it to the left, and it reduces that P50 value. And this is enhancing uptaking oxygen at the lungs. You can also decrease your hemoglobin's affinity. Several of these types of factors will cause your hemoglobin's affinity to decrease. It'll more readily just give up oxygen. And this increases the P50 value, and it favors unloading oxygen to the tissues. So these are two responses. Now, a second mechanism for dealing with the hypoxia that most of you guys are probably more familiar with is the idea that you can just ramp up the hemoglobin concentration in your blood. You can ramp up the number of red blood cells. And this causes you to have then a higher hematocrit. So this would be a higher hematocrit test tube relative to these two. This is good because it increases your oxygen carrying capacity, but it has a potentially bad side effect, which is that it causes your blood to be more viscous. There's more red blood cells in there, and it just makes for thicker blood, and that puts additional demands on your heart just to pump through that thicker blood. Okay, so let's look at how high altitude species and diving species usually deal with hypoxia. So these are typical responses of native high altitude animals. They often have an increased P50. They have that left shifted curve. So these guys are high elevation. They have a left shifted curve. These are vicunias and llamas. And they often have a low to moderate hematocrit. They often don't have high red blood cell count. And vicunias are really, sorry, kicking in the foot. Vicunias are a classic example of this. These guys live in the Andes at 15,000 feet. They're way up there. And they have really high hemoglobin affinity. They're more readily sucking up oxygen from an environment that has low oxygen availability. Now, lowlanders, when they go up to elevation, so this is Emiliano Donadillo, who's one of our colleagues, and he gave a talk here last year on his work on vicunias. And if he goes up to elevation to work on vicunias, he's more likely to actually ramp up his red blood cell production, and he's also more likely to decrease his hemoglobin binding affinity. Diving species, how they deal with hypoxia, they are often encountering, they usually at sea level, they're encountering well-oxygenated conditions. So there's plenty of oxygen, partial pressure to oxygen is good at sea level. So they often have hemoglobin affinities that are similar to terrestrial species. Instead, divers have really high stores of oxygen stores in their lungs and their blood and their muscles, much higher than terrestrial guys. And they often have higher hematocrits. Okay, so with all that said, we are interested in river otters in terms of what happens because they're a high altitude diver. So these guys are a nice model species to look at this because otters are a diver that occurs across a range of elevations, across all of North America. They also have really pretty energetically demanding foraging behavior. The fact that they just chase fish underwater produces really high oxygen demands in their muscles and their tissues. So they have really high O2 demands because of their style of foraging. So our objectives for this section were to essentially create those oxygen dissociation curves for river otters and look at their affinity, as well as just look at their blood chemistry. And then we also wanted to use some of those data to see if we could assess the ability of those Yellowstone otters to, what is really the physiological capacity again, to switch from feeding to cutthroat trout to these deep, open water, novel prey in the sense of the lake trout. So to do this, we looked at otters from two study areas. One was our sea level population is in the San Juan Islands in Washington, and then of course our high elevation population was at Yellowstone Lake. We live trapped otters and from these two populations, and we collected blood samples. And for otter researchers who almost never see otters, we only collect their scats. This provides a nice opportunity to snuggle the heck out of them. And we sure did. I could have showed you about 45 more pictures like that. No, not really. We weren't screwing around that much. Okay, so briefly, methodologically, we used blood gas analyzers to calculate those oxygen dissociation curves, those S-shaped curves. We did hematology and blood chemistry data, and then we also used the hemoglobin concentrations to calculate this thing called a theoretical aerobic dive limit. And this is what is the maximum amount of time you can remain submerged without accumulating blood lactate. And so it's essentially just kind of a picture of what is the dive capacity of an organism. So something like a sperm whale has a really long aerobic dive limit. Something like a muskrat has a pretty short aerobic dive limit. Okay, so we're going to look at some results from this section. So again, our question is, what happens when we have a high altitude diving critter? What are the mechanisms they use to cope with those compounded hypoxic effects? Well, primarily it looks like the Yellowstone guys increase their hematocrit. They really ramp up their hematocrit. They have high amount of red blood cells, hemoglobin content relative to the sea level San Juan Islands otters. And this is actually the highest hematocrit reported in the literature for any study about otter blood chemistry and hematology. Now, that has the problem of the viscous blood, the thickening of the blood. But what they also seem to have is they have high levels of nitric oxide, which is a vasodilator. It's a gas that promotes vasodilation, opens up the blood vessels. And so this may very well be a mechanism for coping with that higher hematocrit. It gives you thicker blood. Well, you can compensate with nitric oxide. And honestly, that's what Viagra does. So anyway, I didn't say that in my dissertation talk. Okay, so I'm going to show you a couple graphs showing here. So O2 capacity is higher for the Yellowstone otters on the black line here. So their O2 capacity is higher, and that's a direct consequence of the fact that they have more red blood cells, higher hemoglobin concentration. But when we look at the affinity, those S-shaped curves, the percent saturation of hemoglobin, the hemoglobin molecules over increasing partial pressure of oxygen, there's no statistical significance in the hemoglobin binding affinity. There's really don't seem very different. And if anything, the Yellowstone guys are slightly shifted to the right, which is that idea of you're unloading oxygen to the tissues, you're promoting getting rid of oxygen, but very little difference. We can see that also when we plot them on a figure with a bunch of other species across a range of elevations. So this is that P50 value, which again is a measure of what your affinity. Do you have a high or low affinity for oxygen over increasing elevations? So the Yellowstone guys seem like they're very similar across a range of elevations, unlike some of these species that have been studied across a range of elevations, such as like foxes or deer mice. Again, vicuña's extremely high hemoglobin affinity for oxygen. Okay, so to tie this back into more of an ecological context, so what does this mean for their diving for lake trout? Well, otter dives typically occur in pretty shallow water and close to shore. There are some deep diving species that actually do have an increased hemoglobin affinity, such as beluga whales, and some of those guys tend to have a high affinity. But Yellowstone otters don't show this. They seem to show really a need to unload oxygen to the tissues, and they don't seem to show a shift towards high affinity. Yellowstone otters also have a really ramped up hematocrit, but really because of the lower oxygen availability at high elevation, all this is doing is buying them about a second and a half of increase of dive time over the sea level guys, the San Juan Islands guys. So it's not really, it's just kind of allowing them to keep pace. So when we kind of look at all this together, it suggests there may not be a lot of physiological kind of room for them to switch to preying on a really open, deep water fish that's just is difficult to catch. Obviously they catch them sometimes, but not a lot. So some quick conclusions from this part. There's no significant difference in the binding affinity of hemoglobin for oxygen between the two populations we studied, potentially due for those just the high energetic demands, high oxygen demands of tissues to the tissues of otters. High elevation otters instead had a higher hemoglobin concentration, and this may be coupled with higher nitric oxide production, possibly to compensate for that idea of increased blood viscosity. Aerobic dive limits, the dive capacities were similar between the two groups, and constraints on diving may really just limit their ability to switch to prey in a deep water environment. Okay, so we are going to go to our third part here, and we're going to extend out to some ecosystem processes and look again at otters providing these nutrient subsidies through their scent marking behavior. They're linking the water and the land. And so in some earlier work we did, we measured growth of shrubs, in particular currants, on otter latrine sites and off the latrines. And we found higher growth in the currants on the latrine was higher in trees on the salmon spawning streams and then reference streams, or streams that didn't have salmon spawning runs. They also found some evidence that the N15 levels, those higher N15 levels, those levels were higher in those larger trees on those salmon spawning streams. So this is foliar Del N15. This is the N15 from the needles. Okay, so one of the potentially kind of, I guess, logical progressions of this is if we know these trees are taking up nitrogen from nutrient subsidies that are made available by some of these animals, and we know that it may influence growth, is there a way we can use it, that we can track changes in nitrogen inputs year by year by year by looking at it in tree rings? Can we go in and look at individual wood and individual tree rings and look at changes in nitrogen 15? And so this has been applied to studies, especially of nitrogen pollution. So looking at changes in levels of nitrogen pollution by looking at N15 in individual tree rings back through time. It's also been used in some of the salmon streams and on some studies of seabird colonies where seabirds bring a bunch of nutrients and nitrogen through their guano and through colonies. In our study, we wanted to try to apply this to the otter cutthroat trout system as well. And so I've shown you several figures with cutthroat trout population data, but they all started here in the late 70s in the high period. If we look back further in time, cutthroat trout have actually gone through a couple periods of low and then a high and then the more recent low. And now if otters during this responded in kind during these years and where their activities were lower and then higher and then lower, this may be reflected in trees at those otter latrine sites because they tend to come back to those things for decades. So that was the idea. Can we measure those changing N15 levels in tree rings back through time? If so, and we compared trees growing on otter latrine with a non-latrine, we wouldn't think we would see a difference between the non-latrine or we would think we would not see that pattern through time in a non-latrine like we would in a latrine tree. So our objectives here were to assess the effects of otter-derived nitrogen on the growth of trees at latrine sites. And because, again, we're hampered a little bit by this lack of historical data relating otter populations to historical cutthroat trout abundance, we wanted to use tree rings to reconstruct historical N15 levels, maybe as a proxy for kind of historical otter activity levels. So briefly, we collected tree cores from spruce trees on latrines and then a reference tree off the latrine within 100 meters of that tree in a paired sampling design. We're comparing each tree. We also did it with spruce or pine as well, but we mostly concentrated on spruce. We just measured tree rings and cross-dated them, and we compared tree ring widths or tree growth with several climate variables, temperature, precipitation, things like that. We then separated, we cut those cores into 5-year segments, and then we measured and analyzed those for percent nitrogen and N15. So results for this part. Well, what we would have expected in terms of tree growth on Otter Homer Drought Severity Index, so a wetter year. So that this positive number, it indicates they're positively correlated. If we look at between spruce trees on the latrines and the reference sites, we see that the latrine trees seem to be kind of decoupled from many of the same climate variables that the reference trees respond to. They're not responding to the same thing. And even more so when we look at the pine trees, in many cases they're responding kind of in the opposite direction to many of the same variables. And so really the take-home message from this table is that these correlation analyses suggest that any differences among trees on our study sites are really driven more by site or tree level variations than climate. They're responding differently. All right, so although the needles from the trees on the otter latrine sites had higher N15, basal area growth was higher on the reference trees. This suggests maybe nitrogen is just not limiting to the trees on the otter latrine sites, or trees may allocate otter-derived nitrogen to some other function, such as reproduction or root growth. We really found no relationship between cutthroat trout and tree ring del N15, and the variation was very high between ring segments. Growth of spruce trees on the otter latrine sites seemed to be decoupled from most climate variables. And overall, given that we found kind of increased growth in shrubs on otter latrines, but decreased growth in trees, this suggests that you may need to kind of investigate multiple plant species or functional groups like grasses versus shrubs versus trees in terms of assessing the impact of this otter-mediated nitrogen transfer at these sites. So I'm going to sum up these 3 parts here, just in case you were napping. And so in our first part, our population part, we saw that river otters responded to declining cutthroat trout primarily via behavioral shifts in their spatial distribution and in their prey consumption. Densities, however, were very low, and it's possible that otter population had already just declined prior to the beginning of our study in 2002. In terms of the physiology study, otters at high elevation seem to use a suite of physiological mechanisms to cope with hypoxic conditions that they face in terms of diving at high elevation. Constraints on diving may limit their ability to capture introduced lake trout. And values of delta N15 were higher in needles of trees on otter latrine sites, which indicates that they've taken up that nitrogen made available by the otters and into their foliage at least. But growth was higher in reference trees. And growth differences among trees on our study sites were influenced more by site or tree level variation than any kind of climatic factors. Okay, so I'm going to have some acknowledgements here for several funding sources, including the UW NPS Research Center. I'd like to thank my graduate committee, especially Merav Ben David, who was my dissertation advisor, and she was the principal investigator on this Yellowstone otter work. A number of great laboratory and field technicians. I'd like to thank the program in ecology at University of Wyoming in the zoology and physiology department. Steve Buskirk and Hank Arlo, who was a co-author on that high altitude diving. I'd really like to thank Bob Landis and Bill Wengler, who were filming or doing work out here prior to our work, and so they really kind of showed us where otter latrines were at the beginning of the study. They were absolutely instrumental in getting us up to speed there. And then tons of folks out in Yellowstone National Park who provided logistical support and made the whole thing possible. So, questions, if anybody has any questions.
So the lake trout spawn in the fall, which doesn't cycle with the reproduction of the otters. In this lake, in Jackson Lake, there's some very specific areas where we know where the lake trout spawn. I was curious whether there might be any latrine sites near lake trout spawning grounds in Yellowstone.
Yeah, so in West Thumb, where there's some lake trout spawning grounds, we do have some great otter latrine sites. There's 2 or 3 in there that are really some of the best ones that they continued to come back to all the time. So they're definitely there near the lake trout spawning grounds. Now I don't know if that they eat those lake trout in the fall. I know that otters have latrine sites there, and I know that they eat cutthroat trout and suckers around there. And actually, that's actually a good question, because I show you those 4 or 5% that they eat the lake trout. A couple of those do come from. Those tend to come more from that area that's near those spawning grounds. Now, a thing that I hope we get a chance to do someday, or somebody does, we don't know what those guys are doing, what the otters are eating in December. So, maybe they eat lake trout then. There's plenty of documentation of them eating cutthroat trout and suckers and pictures of that and all the stuff Bob Landis filmed, but I don't know what they do then.
You mentioned in some of your analyses that you found evidence for inbreeding. Did you do any parentage analyses to try to get direct evidence of inbreeding?
No, we didn't do parentage analyses in there. That's something that we might do before this thing goes to publication. Actually, that's a good thing. Yeah, it was strictly just a test to see what the inbreeding coefficient was, and it showed some evidence of it, and slightly decreased heterozygosity, which I wasn't too surprised by. But yeah, that's a good point. Yeah. Somebody else over here thought, man, people want to watch basketball.
You see that the otters, higher elevations have higher viscosity in their blood.
Yeah, well,
Do you know, your knowledge, is that through a human being also?
yeah. So if you guys came from California, then yeah, you did. You increased your red blood cell production. You have thicker blood. Now, what's kind of interesting about that question? That's a good question. Those studies of Tibetans have higher nitric oxide production, as do Andean highlanders. So those guys also have that, which may be a compensation for increasing, because especially the Andean highlanders actually have increased red blood cell production. So they do kind of what we think of as an acclimatization response from going from California to here. Tibetan highlanders actually have increased red blood cell counts. Or what did I say? Andean highlanders have that. Tibetans don't. They're pretty similar to sea level people. But Tibetans, even without that, they increase their they still have more vasodilation just to pump blood through faster to get oxygen out to the tissues faster. But yeah, you should if you acclimatize.
The sea level population of otters that you studied, those river otters?
Yep, they're river otters, but they're coastal river otters. So in the San Juan Islands, they go in and out of the saltwater habitats as well. But they were river otters, yeah. No, good question. Yeah, and it was really just because my major advisor has a collaborative partner who does a bunch of research out there, and so similar to out here, they know where the auto trains are and they know where to capture them and stuff like that. But yeah.
Notice from a couple of your slides that it looks like otters move a fairly good distance. And sorry if I missed it, but do you have the whole estimate of home range of individual otters?
No, I'm sure that they leave this population. It's an open population. They may easily go up to Trout Lake and things like that, and I don't know where they go. We didn't feel comfortable in just genotyped information where they were in like really calculating what a true home range for those guys are. I could tell you that they moved from. We had one female that moved from the southeast arm of Yellowstone Lake, which is the far end of that lake, in those maps I showed you. I don't know how to return this thing to the, unless I go like that. And up to the Yellowstone River, so I mean that gal moved. I know this is the worst way to do this actually. No one likes this, including me. But just for those of you that don't know, oh my God, where's the map? So she went from down here to up here. So, but yeah, they move. And I mean, Nathan Varley and Bob Landis, when they were following around filming them, they saw them move 6 miles and then back up the river 7.5 miles in 4 hours. So they move.
I'm praising those guys, I'm tracking them.
So
What
I would
preys
say the
on
main
those
thing
guys?
that will prey on them would be coyotes, but wolves eat them every once in a while. But the main thing that they get hit by is coyotes, especially in the winter. And if any of you guys have seen films of them, the coyotes will come in and steal trout for them when they're going into the so you know, another unique thing about Yellowstone Lake is it has those thermal features that open up holes for them to fish, and the otters will go in there, catch fish, come back up, and a coyote will come along and steal it from them. Sometimes they attack them. And there was a mother with pups up in Trout Lake, up in the northeast part of the park, that got killed by coyotes. And I talked to some trail crew people who saw a wolf running around down a trail with the otter hanging out of its mouth. Now, what I will say is otters really don't. That's not a big problem for them. I mean, they can get in the water, and so for the most part, otters aren't facing the same thing as a lot of those guys. If you're we were talking about this at the barbecue, though. If you're a giant otter or something that lives in South America and there's piranhas and crocodiles and things like that, then it's a whole different ballgame. But that's a great question, yeah. I would say the main things would be like those coyotes and wolves.
Any more questions? Thank you, Jamie.
Thanks, guys.